Good day everyone, and I welcome you back to our channel today. Today we're going to discuss the characteristics of bacteria, and this one is very important to note. Now, why do we need to study all these different characteristics wey bacteria get? We need to understand say bacteria is ubiquitous, meaning say if you find them for everywhere, every environment, bacteria don't conquer am. Now we get the kingdom wey we dey call kingdom Monera, and this kingdom na only bacteria and archaea dey inside this kingdom. And bacteria and archaea dem be wetin we dey call prokaryotic cells. Now prokaryotic cells na primitive cells, they no dey as specialized as the other ones wey we dey call eukaryotic cells. Now e dey important to understand, we go look at some of the characteristics wey bacteria dey get. Now the first characteristic wey bacteria get na say bacteria na wetin we dey call simple microbial cell. So na simple cell, and most of the time na unicellular cell, meaning say na one single cell. Now this single cell fit dey together with other cells, e fit appear in a community, e fit appear in pairs or even as four cells. But the important thing to note na say each single cell na a different entity on its own, and e dey run all the life processes by itself. This one dey important to note. So the bacterial cell na simple unicellular cell. Another important thing to note na say the bacterial cell get wetin we dey call peptidoglycan layer. Now this peptidoglycan layer na the cell wall wey the bacteria get. That cell wall dey made up of wetin we dey call the peptidoglycan layer. We go still shoot a video where we go discuss this peptidoglycan layer in details, look at all the things wey dey inside this peptidoglycan layer. Now this cell wall dey very important because the cell wall dey give the bacteria strength, e dey give the bacteria shape, e dey give the bacteria size. Now e dey also help the bacteria to control the things wey dey enter and wey dey comot from the body of the bacteria. We know say any target, maybe na drug or anything, if you target am at the cell wall, if you fit destroy that cell wall or break am for one side, e go lead to the death of the bacteria. So the cell wall dey very important as e dey run many processes for the bacteria, and this one dey very useful to know. Another important thing to know na say the size of the bacteria. We dey measure size of bacteria using unit of measurement wey we dey call micrometer. Now micrometer dey very small, and the way to understand wetin micrometer be na say if you take your ruler wey we dey use rule things, we dey use, right? If you take that ruler now, look for that ruler, you go see say many rulers dey get centimeters, other rulers dey get meters. Now 1 meter for a ruler, if you divide that 1 meter into a million places, one of that na wetin you dey call micrometer. And this is why our bacteria na wetin we dey call microscopic organism, meaning say you no fit see am with your naked eye. You go only fit see am if you employ the use of the microscope. Now the size of bacteria dey range many times from 2 to 5 to 7 micrometers. Really small, but this one dey important to note. Now bacteria also dey lack true nucleus. Because they be prokaryotic cells, they no be eukaryotic cells, they no get nucleus, they no get any membrane around their nucleus. Rather, wetin bacteria get, wey all the other prokaryotic cells get, na wetin we dey call nucleoid region. Now wetin be nucleoid region? Na area within the bacterial cell where all the genetic material been gather put for that area. So they go dey for that area. Now the bacterial cell get wetin we dey call single circular chromosome. Na one chromosome dey inside the bacterial cell, one single one, and e dey circular, na giant one, but e go coil very well into that area where we dey call the nucleoid region. So this nucleoid region dey carry all the or most of the genetic content of the bacteria, the genes, the traits, all the characteristics wey the bacteria dey use run the different processes go dey inside that nucleoid region. Now if you dey view the bacteria on top microscope, you go notice say that nucleoid region go dey darker. We dey talk say e dey stain differently because e go dey much darker than the other regions of the cell. This one dey important to note. Now apart from the nucleoid region wey be a chief characteristic of bacteria, many bacteria dey get wetin we dey call plasmids. Now wetin be plasmid? Plasmid na DNA wey dey different, wey be say e no dey for that nucleoid region, but e dey for any other region of the cell. So we dey sometimes refer to them as extra chromosomal DNA. Now extra meaning say e no dey along with the nucleoid region. Chromosomal na say e no dey for that chromosome. So extra chromosomal DNA, plasmid, na example of extra chromosomal DNA wey dey carry many. And plasmids dey important too because they give particular characteristics to bacteria. If they carry those plasmids, the bacteria go get some characteristics wey other bacteria wey no carry that plasmid no go get. So this one dey important to note. Now, reproduction for bacteria na through a process wey we dey call binary fission. Binary fission, and this one mean say a single bacterial cell wey be parent cell go divide to form two daughter cells. So a single bacterial cell go divide to form two daughter cells, and those two daughter cells after a while they go divide too to form other daughter cells. So this time we dey sometimes call am reproduction time or turnaround time for the bacteria to move from one parent to two daughter cells through the process of binary fission. This one dey important to note. Another thing to note na say apart from binary fission, sometimes bacteria dey go into wetin we dey call conjugation. And wetin conjugation mean na say two bacterial cells go come together, they go dey lie side by side, and they go exchange a lot of their genetic material from the different nucleoid regions or from their different plasmids. They go exchange many different genetic material. Now this one dey important, and it's also good because e dey help bacteria to vary the genetic trait and the genetic content wey dey inside the bacteria. Because if this one no happen, na only the same genetic material go dey move from one generation to the other to the other. So conjugation dey occur once in a while for bacteria. Now, very many different examples dey of bacteria wey we know. Staphylococcus aureus, Escherichia coli, Pseudomonas aeruginosa, Klebsiella species, many different bacteria dem dey, and they dey useful for many things. Some of them dey cause illnesses, so we dey look at them as pathogenic or disease-causing organisms. Some we dey use them produce antibiotics, others we dey use them produce vaccines. We dey use them for different things wey go benefit human beings. So this one dey important. Now we also know say bacteria dey get both internal and external structures. These internal and external structures dey important, and we go shoot a video where we go discuss in detail all the different internal structures wey dey bacterial cell and the external structures, and wetin these structures dey do, wetin be their usefulness. We will try and know all these things for the video wey dey come up. Now, thank you for listening to this video, and please like our content, share am, give your friends, and have a wonderful